Hello students, we have already completed vegetative and asexual reproduction, and now we are going to start with sexual reproduction in fungi. What will be the most important difference between asexual and sexual reproduction in fungi? Asexual reproduction involved only one parent, but for sexual reproduction, two parent fungi will be involved. These two parent fungi are called as compatible mating types because they are genetically compatible to mate with each other. They cannot be any random two fungi. Two parents are involved. These two parents are called as compatible mating types, denoted as plus and minus. Either the gametes or hyphae of the two compatible fungi will mate with each other. Either gametes or hyphae of two parents mate. Mate matlab bring about sexual reproduction. It is completed in 3 steps, and the 3 steps are: number 1, plasmogamy. What happens in plasmogamy is, let us say these are the two gametes from the two compatible mating types. They are denoted as plus and minus. As you can see, the nuclei that both of them will be having will always be haploid, never diploid. Gametes are always and always haploid. Rule. That's a law. So in this step, the two gametes are going to fuse together, and what we'll achieve is this situation. Since both the cells have fused together, the two nuclei have arrived in one cell. The wall has dissolved between them, and the situation that we have right now is n plus n, not 2n. Okay, let's write down over here, not 2n. It's not 2n yet. This cell with the two haploid nuclei is called as a dikaryon, wherein di means two, karyo means nucleus. So that's the first step, fusion of gametes or fusion of hyphae alternately. This will be the situation, plasmogamy. We achieve a dikaryon. In the second stage, what is happening in this second stage? The two haploid nuclei have fused together to form one nucleus, which will be 2n, which is called as the zygote. Gametes are always haploid and zygote is always diploid. That's the law. This is the way. So because this is a fusion, this is a game of nuclei also being called as karyon. What will this step be called as? The second step will be called as karyogamy. Karyogamy. Name of the second step of sexual reproduction in fungi. Finally, we come to the third step. What is the fate of the zygote in humans? In humans, once the diploid zygote is formed, the first division that it will undergo will be mitosis. Why does it undergo mitosis and not meiosis? You know, cell divisions are of two types, mitosis and meiosis. Mitosis being equational division. When a 2n cell divides, the second cell that will be produced will be a 2n cell only. Okay, when that cell divides by mitosis, 2n will be formed. But 2n cell undergoes meiosis, what is going to happen? When a 2n cell undergoes meiosis, the 2n ploidy will be reduced by half. So 4n and nn cells will be formed. Mitosis, meiosis, two cell divisions. If a 2n cell divides by mitosis, 2n cells will be formed. And when the same 2n cell divides by meiosis, 4n and nn cells will be formed. Since the ploidy of the cell is maintained in mitosis, it is called as equational division. And since the ploidy of the 2n cell is halved in meiosis, this is called as reductional division. In humans, what is the fate of this zygote? In humans, the diploid zygote that is formed undergoes mitosis and not meiosis, because the ploidy of the fetus is supposed to be 2n. We are 2n. Our every cell is 2n in nature. But for a fungi whose ploidy is never diploid, their ploidy is haploid. So what will be the cell division that the zygote will have to take now? It will not undergo the process of mitosis, it undergoes the process of meiosis. Once the diploid cell undergoes meiosis, what is going to happen? 2n, it will give rise to 4 haploid cells. In this manner, ploidy will be halved from 2n to n, and then once these haploid cells are formed, may be released as spores. So these are the 3 steps of sexual reproduction in fungi: plasmogamy, karyogamy and meiosis. The 5 classes of fungi that you have learned are oomycetes, zygomycetes, ascomycetes, basidiomycetes and deuteromycetes. These are your lower fungi and these are supposed to be your higher fungi, isn't it? Do all of them undergo sexual reproduction? Sexual reproduction. In oomycetes, sexual reproduction is present. Zygomycetes, sexual reproduction is present. Ascomycetes, present. Deuteromycetes, sexual reproduction is absent. In fact, that is the reason why they are called as fungi imperfecti. We will learn. Sexual spore. The sexual spore that is produced in case of oomycetes, the sexual spore is oospore. For zygomycetes, that will be zygospore. Ascomycetes will give you ascospores. Basidiomycetes with spore, basidiospore. And deuteromycetes with spore, deuterospores. No, they will not produce sexual spores since sexual reproduction is absent in deuteromycetes. Absent. So since in these 4 classes sexual reproduction is present, all the 3 stages will happen: plasmogamy, karyogamy and meiosis. However, apparently for these lower fungi, plasmogamy is immediately followed by karyogamy. Why I am saying is because in the higher fungi, karyogamy is delayed after plasmogamy. Why? Why this process of karyogamy is delayed after plasmogamy in the higher fungi? You will understand when we are doing higher fungi in detail. Here is what happens in basidiomycetes. Can you tell me what has happened over here? So in basidiomycetes, these are the 2 compatible hyphae, plus and minus. The nuclei differences you can see clearly. These 2 hyphae are fusing together. The hyphae that will grow will keep on having 2 nuclei. Even as it grows, all the cells are going to have 2 nuclei in this manner. This is called as the dikaryophase, wherein the cells have a dikaryon. Then what is going to happen is these hyphae are then going to grow. Let's say these are the hyphae. They will grow, clumping, and what will be formed will be this. This is your mushroom. If you check each of these cells inside this mushroom, you will find that every cell has n plus n within this. These are the gills. I've taken a gill here, this section. This is the gill. So you will find these hyphae over here. These are the hyphae, and these are the terminal portions of this hyphae. If you check one of these terminal end of this hyphae, see dikaryons, you can see clearly. So you see in this terminal cell, there is also a dikaryon situation. After all of this journey of dikaryophase, after plasmogamy, this is the step of plasmogamy here. After a prolonged period, as you can see, finally in the terminal cell, the nuclei are going to fuse together to produce a 2n nucleus. This will be the zygote. And then there will be the process of meiosis, wherein within this cell, 4 nuclei will be formed. And then, see what has happened? Nuclei have come out of this and settled on this N. These will be your basidiospores. So this diagram is a representation of this sentence, that in the higher fungi like ascomycetes and basidiomycetes, the karyogamy is delayed after plasmogamy. But in the lower fungi, the plasmogamy is immediately followed by karyogamy because they are very lesser fungi. They don't need to do nuclei. They will immediately fuse their nuclei to carry out karyogamy.
महादेव के नाम पे सब्सक्राइब कर दें।